What I presented today were experiments showing that the mechanical strands that are developed by tumor growth can activate biochemical pathways that are tumourigenic, that are dangerous, in fact, in healthy tissues surrounding the tumor. So this is kind of mechanical contamination of the healthy tissue by the tumorous tissue, which is unusual because classically people and research found that molecules has to be secreted, produced by the tumors, and diffuses into the healthy tissue, and it can eventually activate tumourigenic pathways and tumorigenesis in healthy tissues thanks to these signals. And in our case, what we found is that in addition to these biochemical signals that are associated to molecules that diffuses, there are also mechanical signals in terms of the pressure developed by the tumors compresses healthy tissues, and this compression activates tumourigenesis in these tissues. So it transforms the healthy cells into hyperproliferative cells, cells that begin to divide too much, and eventually, in certain cases, it leads to also tumourigenesis in these cells. Well, it's fundamental research, so it means that we found a new way for which tumor progresses. So it means that it has also new targets to fight against with pharmacological approaches, chemical approaches, and eventually chemical treatment approaches. But this is not yet what we are doing. I mean, it's really fundamental research. It opens to new possibilities to cure cancers, hopefully in typically 10 years or something like this. We are not thinking this process as purely mechanical. What happens, in fact, in the cells, healthy cells that are compressed, is that the mechanical deformation, in fact, changes the biochemical state of certain proteins, because it's easy to change the conformation, the shape of the protein in response to mechanical strain. And changing the shape of a protein or a conformation of a protein can eventually activate this protein and then activate a classical pathway which is biochemical. So the goal would be the goal will be to treat chemically this process, meaning that you could try to inhibit chemically the mechanosensitive pathways and see if it helps to fight against tumor growth. So you can use chemicals against mechanical induction because mechanical induction mixes mechanics and biochemistry. So you will try to inhibit the mechanosensitive pathways. The technology is a bit unusual. In fact, we are in fact a consortium of different teams in Paris, center of Paris, and we aggregated around Institut Curie, which is an institute in which physics and biology cross-talk together very much since more than 10 years now, 20 years. And in fact, what we did within this context is that to use tools of chemists who produced magnetic liposomes. And what you can do is to inject these magnetic liposomes into the blood. And you can put a small magnet in front of the organ of interest you're working on. In our case, it's the colon. And this, in fact, helps the liposomes to escape the blood flow, and to what we say is that it extravasates. It goes out from the blood, and then it can incorporate into the colon tissues. and it's very stable for weeks to months in mice. And then you can use the magnet, which is localized in front of the colon, also to apply a mechanical strain to the colon, which is magnetized. And luckily, in fact, we could mimic quantitatively the tumor growth pressure thanks to these tools. So we could apply for weeks to months what is, in fact, the pressure developed by a tumor, but without a tumor. So we're sure what we do is directly mechanical. I mean, it's really the mechanical pressure that activates all these tumourigenic pathways, independently of any biochemical molecules that would be secreted by a tumor. So the tools we are using were designed to test the role of this parameter, this mechanical parameter in tumorigenesis, yes.